Hi everyone, Michelle Formiglio, and I'm going to be narrating this PowerPoint, which is chapter 22, the respiratory system. This is going to include anatomy in the first part mainly, and physiology in the second part. It's approximately 2 hours, so feel free to take a break somewhere in between. But make sure you take some notes along the way and refer to your textbook. So this is Human Anatomy and Physiology by Marib and Hohen, and this is the 11th edition. So we're going to jump right in. So our first slide here is a discussion of functions of the respiratory system. So there's a little video clip here you can watch if you want to called Meet the Lungs. So feel free to do that, pause this or come back to this and feel free to do that. So then the first thing we need to talk about is what is the job of the lungs. The main job is gas exchange, right? So we need to get oxygen into the bloodstream and we need to get carbon dioxide out of the bloodstream. And besides that, the various structures of the respiratory system are involved with taste and smell and, a little bit of defense of the body, a couple other things. But yeah, so those are the main functions. And sorry, I'm just going to be kind of looking here and there. So page 819 is going to lead us to this bottom portion of this slide, which is the processes of respiration. So the four main terms, and we're going to come back to these, but they are pulmonary ventilation, external respiration, internal respiration, and transport. So just as a heads up, pulmonary ventilation just means breathing. We're going to discuss, how we do this, what we do with the carbon dioxide, that we're carrying. What are some terms that we use to describe different conditions that we might have. Basically an overview. We're not going to go really deep into any one topic because we have a couple hours to do an entire chapter. But yeah, so we're going to be talking about all of those, but pulmonary ventilation in general just means breathing. Okay, so basic function or basic structures of the respiratory system. This is where we're going to start. Now, if I were you, I would begin a list, kind of numbering the pathway of air through the respiratory system. So I would start with nose and I would work my way down, pharynx, larynx, trachea. You'll see that on one of my slides, but it's a good way to study. You also need to make sure that you're able to tell me the difference between conducting zone structures and respiratory zone structures. So the upper respiratory system is going to pretty much be everything down through your voice box. After the larynx, it's considered lower respiratory. Trachea, bronchi, lungs, alveoli. So conducting zone structures are areas where there is no gas exchange, no air exchange. The whole point of a conducting zone structure is just to get the air into the body, get it, kind of moistened, humidified, cleaned, warmed up on its way down, so that by the time it gets to the terminal bronchioles and alveoli, which are the very, the functional component of all these structures, that there's the ability to do safe gas exchange. All right, and so this picture, you should make sure that you can label all of these structures. This picture is on page 820. So main structures, I'm going to be referring back to this particular picture often. The way that this presentation is lined up, I don't think I can sort of scroll back and forth between my slides. So if I were you, I would have this pulled up, page 820. And, unless of course you have this printed out somewhere. But so I'm going to refer back to this picture a few times. Sorry, I'm trying to get comfortable here. All right, and so again, major components are listed here. Upper respiratory system is going to involve nostril, nasal cavity, oral cavity kind of, pharynx, and larynx. And then after larynx, we're now entering lower respiratory system. Trachea, the primary bronchi, secondary, tertiary bronchi, all the way down to alveoli and the lung itself. All right, so let's start with the nose. Picture on page 821 has some of our nose structures. So the first thing is the function of the nose. The nose provides an airway for respiration. That's what it does. Within the nose, we have some facial bones, cranial and facial bones, that are going to be involved with, obviously the structure, but then we have these nasal conchae that are inside of the nasal cavity. And their job is going to be kind of to swirl that air around within the nasal cavity. And then as it's sort of swirling around, recall what those nasal conchae look like. And if you're not sure, then take a couple minutes to look at the picture on 821. And, recall their job, which is, again, air comes in, and as it passes over the mucous membranes of the conchae, then you're going to see that air warming up, getting humidified, and also getting filtered by the goblet cells. The goblet cells aren't really filtering it, but the goblet cells that are present in that location are going to create mucus that's sticky, and it's going to allow for us to snag that bacteria or whatever germs or dust or debris that might be coming in. So, yeah, it's going to filter the air, clean the air, warm it up, and then the other thing that the nose can do is it can provide a resonance chamber for speech, which really just means, you have kind of an open area in here. Sinuses have the same type of role, and that is going to allow sound to amplify. So I always think of like acoustics. The nose also houses olfactory receptors. I'm going to try something here. Olfactory. Oops, it looks like I crossed it out. I'm sorry. Let me erase that. Okay, let's use yellow. So olfactory is a term that you should make sure you understand and know. Olfactory means smell. So make sure you understand that. There are olfactory receptors at the top of the nasal cavity. All right, so in general, we have two regions, the external nose and the nasal cavity. So let's go on to external nose first. This picture is on page 821. It's similar. So really, the structures here, root, bridge, apex. The root of your nose is the area right between your eyes. So the root of a structure is not always the widest part of a structure, but it's kind of a part where the structure, that's almost like where it would grow from almost. So this would be the root. The bridge of the nose is also known as the dorsum of the nose. This is the part that people tend to break. And then the tip would be also known as the apex. A term that we use for nostril is nair. So you should know that nair and nostril are the same thing. And that's pretty much it. Okay, so looking at the nasal cavity itself. So the nasal cavity is composed of a septum, a roof, a floor, and a nasal vestibule. So you should be looking again on page 821. And I'm moving ahead. This is slide 7 now. So the septum of the nose is what's going to divide the nasal cavity into left and right. So think of interventricular septum, interatrial septum, anything that's, dividing something. So this septum that divides the nasal cavity is made of two bones that should be a review for you. One is the vomer and one is the ethmoid bone. So your vomer bone was kind of a pointy bone at the base of your, sort of at the bottom of that nasal cavity. And the ethmoid bone is going to be really responsible for a lot of those nasal conchae. So we're talking about, the septum is medial. And then we also have cartilage. The roof of the nasal cavity. Okay, so we're talking way up, kind of below the frontal lobe. That is also ethmoid bone and then sphenoid bone. So if you recall, the sphenoid bone looked like a bat-shaped bone, and sort of sitting in the middle of it would be pituitary gland, if that gives you a better idea of where we're talking here. So that's going to be the roof of the nasal cavity. The floor of the nasal cavity is shared with the top of the mouth. And so that structure is formed from bone and from fleshy material. So the hard palate and the soft palate are the two structures there, and we'll discuss those in just a minute. This is not easy to do. And then the last part here, the nasal vestibule, I'm not so much worried about, but I do want you to understand this term, vibrissae, is hairs. I don't use it very often, that term, so I wouldn't stress too much about it. All right, so now we have this term olfactory again, which is smell. So at the top of the nasal cavity, we have our olfactory mucosa, and within that olfactory mucosa are smell receptors. They're going to gather, they're really going to respond to or receive chemical information, which is going to be in the air, and that information will be sent to the olfactory nerve and eventually get processed in the brain. So then we have our respiratory mucosa. So the other mucosa involved is going to be specialized for conditioning the air. So the first one, the first thing you need to know is the type of tissue involved, and this is pseudostratified, ciliated, columnar epithelium. That type of tissue is going to have mucous membranes. It's also going to have cilia. So the mucous membranes are going to trap secretion, or going to trap waste, trap junk, and the cilia are going to help move that junk along to remove it from the respiratory tract. And so there's your tissue type right here. And then we have secretions, mucus secretions, which I already kind of mentioned. So then we have a couple of antibiotics, natural antibiotics. The first one here is called lysozyme. And if you split lysozyme into two pieces, okay, that did not work. Sorry about that. Erase. Split lysozyme into two pieces. Lyso means destroy, and zyme means enzyme. So lysozyme is a destroying enzyme, and that can be deployed when there are bacteria around, and it can destroy the bacteria. Defensins are somewhat similar. And so we already talked about air that is inspired or breathed in becoming warmed and humidified by the capillaries in those mucous membranes or below the mucous membranes. And also what can happen in this region is that sensory nerves can trigger sneezing. So that's almost like a little reflex that can happen there. All right, so let's move on to the nasal cavity, a discussion of these structures inside. This is on page 821, still moving into 822. Yes. All right. So those nasal conchae, I already mentioned a couple times. So there are three sets, superior, middle, and inferior sets. I already discussed what they do, but it says it again, so we'll talk about it. So they are extending from the lateral walls, protruding medially, and job, increase the ability of these tissues to, of the air to kind of circulate around, filter, heat, humidify the air so that as it goes down into the lung, gets to the lowest parts of the lung, it's conditioned a little bit already. So the nasal conchae are these little curves, right, made by ethmoid bone and some structures in the nose. And then after all those curves, and I'm going to show this on the next slide, like sort of at the base of those curves, there's a little groove, and that's called the nasal meatus. And it almost looks like a little tunnel. If you remember, meatus was a tunnel. If you think of your acoustic meatus or the urethral meatus, that's that. So I'll show you on the next slide. Okay. These guys right up here. So it's supposed to make a circle somehow. Hold on. Yeah, that's not working. Oh, it's not. Okay. Erase. Sorry about that. Okay. So what we're looking at here, these are the nasal conchae. Let me highlight this. Nasal conchae. Sorry. Okay. Nasal conchae right here. Okay, so these are these curved structures, nasal conchae, nasal conchae, nasal conchae. And then underneath of the nasal conchae, we have the meatises, which are like the curve, the underspot, the underneath spot, yeah, after the curve. So they're little grooves are considered. Here's your hard palate and your soft palate that I had mentioned. So your hard palate is here and soft palate is here. And we'll discuss those a little bit more soon. So these are like the top of the oral cavity, so that's why we use the word palate, but they're also the bottom of the nasal cavity. Our next slide is going to talk about sinuses, so I'll point those out to you right now. Frontal sinus is one of our sinuses, the sphenoid sinus. I don't see our other two sinuses, but I do have a picture on the next slide, so let's look at that now. All right, so these sinuses are called the paranasal sinuses. So para as a term means beside, or alongside, beside the nose. And so looking at all of this, you can see the names: frontal, sphenoid, ethmoid, maxillary. So frontal refers to the sinus in the frontal bone. Sphenoid, ethmoid. Sphenoid and ethmoid are the ones where you kind of like squeeze your bridge of your nose when you feel like you have sinus pressure. That's those guys. And then maxillary is right at the maxilla. So the job of these sinuses, the big one is to lighten the skull. Remember, our brain is really, really, really heavy. It's very vascular, and a couple of air spaces in the brain, not in the brain, but in the skull help with the weight. So they make some mucus, which is going to help clean the air, and they are connected to the nasal cavity. So they also help warm and moisten the air, and your sinuses are really good amplifiers of your sound, of your voice. All right, moving on to our next part. So again, if you're writing some notes down, so I would put like 1, nose. 2, pharynx. So we're just following, wow, sorry about that. Okay, so, and then you'd proceed down all the way. You're following, picture following one drop of, not one drop of air, but like one molecule of air moving through. So this would be, you'd call this like the pathway of air, something like that. So our second step is the pharynx. The pharynx has a few different parts to it, and I'm actually going to erase this because it looks like a child wrote it. So, but I think you get the point. There we go. Excuse me. All right, and so the pharynx is also known as the throat, and if you look, you're going to see 3 parts. We have the red part, the blue part, and the green part. The red part is known as the nasopharynx. Naso referring to nose. The blue part is the oropharynx, a little further down, and the laryngopharynx is the lowest part right in front of the larynx. So those are the 3 main regions. They're not super clearly defined, definitive as far as where one starts and the other begins. But, this is basically when you're looking at the word parts, that's what you see. You see nose, behind the mouth, behind the larynx. So the throat, we're talking about an area that's about 5 inches and connects the nose to the mouth and the larynx. All right, so now we'll talk a little bit more about what's going on within those structures. And now on slide 13, and in your textbook, a good page for all this is 823. Okay. All right, so let's look at that. The soft palate Okay, so here's your mouth, right? This is your nasal cavity, and here is the bony part of the roof of your mouth, which is the hard palate. The soft palate is this region right in here. And on this slide, it's going to, or on this picture, it's going to be right in here. All right, so the soft palate hangs at the back of the mouth, and at the very end of the soft palate is a little guy that hangs down, and it's known as the uvula or uvula. So when the mouth is closed for swallowing, the soft palate and uvula or uvula close, and or they drop, and that's going to help prevent that food from going down the wrong way. I'm sorry, I just said that backwards. The soft palate and the uvula close the nasopharynx when swallowing. So really what the point of that is to prevent food from going back up into the nasal cavity. So we also have these little guys. Let me erase these and let's talk about the eustachian tubes. So I'll do this in purple. So right here, that little guy, boom, is the eustachian tube. And I'm sorry I can't make this larger for you, but this picture is not in your textbook. I'm not really sure why they don't have this. Those tubes are kind of important. They must be covered in a chapter with the senses and the ear. But anyway, so this tube that connects the middle ear with the nasopharynx and nasal cavity is known as the eustachian tube or auditory tube. Sometimes it's called a pharyngotympanic tube. But that tube's job is to equalize the pressure and drain excess fluid from the ear. And that's the goal. Unfortunately, right at this particular part, there's also a tonsil called the pharyngeal tonsil. And pharyngeal tonsils are also known as the adenoids. And so a lot of times kids might have issues with a lot of ear infections. They have to have their adenoids removed and have tubes put in. They're not really tubes. They're like a little tiny valve. So they have to have like a valve put in and remove their adenoids because so many infections can start causing blockages to the passageways, and also they can kind of clog the ears. So a lot of kids might have delayed speech because they could have so much blockage in their ears that they might not be able to hear it, to hear speech and learn. So let me erase this and go back to this slide over here, or this picture on the right, and I'll try to highlight those tonsils. So the first tonsil here is this guy, and this is known as the pharyngeal tonsil or adenoid. Our second tonsil is known as the palatine tonsil, and that's right there, palatine. And so the palatine tonsils, like if you look in the back of your throat, they're on either side of the back of your throat. And then you have a little guy down here known as the lingual tonsil. So these are on 823. So they are on the slide. Slide 12, or I don't know if it's 12 or 10 that has that picture. But anyway, 823. And so lingual refers to tongue. When you say someone's bilingual, it means they have two tongues or two languages that they can speak. All right, so then we have our larynx. Larynx is also known as the voice box. So again, if you're doing your little list of steps, so you'd have 1 would be the nose, 2 would be pharynx, 3 is going to be larynx, 4 is going to be trachea. So that's next. All right, so the hyoid bone right here, this little guy, you should have learned when you learned bones. So the hyoid bone, oops, U-shaped, I actually meant to do this. U-shaped floating bone in the neck. It's attached to the other structures through ligaments, But the hyoid bone is extremely important for the structure of the larynx and the connection to the tongue. So the larynx is not very large. It's only a couple inches. It takes up the span of about 3 vertebrae. And its function, its job is to route food and air to the right tubes and voice production. So the tubes we're talking about are the trachea, and behind the trachea, which is the esophagus, which will run behind the trachea. So this guy, the epiglottis, is the big one as far as, you know, he's the guy who routes the air in the right direction, or routes the food or air in the right direction. So let's go forward a little bit. We'll talk about the structure of the larynx a little bit more. This is page 825. So here's our larynx. There are 9 cartilages in the larynx. I wish I could enlarge this for you a little bit, but again, this program isn't letting me right now. But at any rate, so there are 9 cartilages in the larynx, lots of connective tissue. What we're looking at here, shoot, I wish I could go back. So refer to your book, page 825, and I want you to pay attention to or look at the label that says epiglottis. So I want you to look for the epiglottis, and in this picture, this is kind of a top-down view, but we're looking at this structure right here. Now what the epiglottis is going to do is it's a flexible piece of tissue that's made mainly of elastic cartilage. And elastic cartilage is a very flexible cartilage, and it's like, you know, if you can put your ear into a little ball and it'll spring right back, that's elastic cartilage. The epiglottis is also elastic cartilage. So the epiglottis is, it looks like a leaf almost, the shape of it. And its job, like if this hole is the glottis leads straight down into the trachea. And you can see it in this picture up here, right here. So this hole is the glottis. And the epiglottis covers this hole when food comes in the situation, because we don't want food to go down into the glottis. So epi means upon. So the epiglottis sits upon the glottis when food enters the larynx area, so the laryngopharynx. So once that's covered, air can't get down anymore. Instead, the food comes in and gets routed kind of posteriorly and down the esophagus. And so the epiglottis is really just kind of like a valve almost that's going to push, doesn't push the food, but it blocks off one pathway so that the food has to go the other way. So the epiglottis covers the glottis, and then we have a few other cartilages. There are 2 that you need to know the names of. So again, if you want to flip back to the slide prior, on 825, there are the 2 that I'd like you to know for lecture are known as the cricoid cartilage and the thyroid cartilage. The thyroid cartilage is the Adam's apple. Oh, the Adam's apple is part of the thyroid cartilage, so it's a large shield-shaped hyaline cartilage piece. So that's you know, your thyroid is just below. So that's a really large, you know, that's the biggest one. And then the cricoid cartilage is a ring shape, and it's kind of like sort of below all of that. So if you look back, I'm sorry, if you look back to that slide or to that picture, you'll see cricoid cartilage. So all of these cartilages, epiglottis is here. Let me do my. So we have epiglottis, the E, and elastic. Epi is elastic, and these 2 cartilages are hard hyaline. Sorry, I'm not good at this. Okay. So then the other cartilages that are involved, you don't need to know the names for them for right now for this class. And then there are other cartilage. Those other cartilages have different names, arytenoid and a few different names. Those are involved with the vocal cord. So I'm going to highlight these vocal cords in blue here. this sort of middle area right in here, these are your vocal cords. And let me see, this picture is, it's not a drawing, it's an actual picture in your book on 826. But it's pretty much the same exact thing. So those are your vocal cords. And then sometimes we call them your true vocal cords. And then I'm going to highlight in orange. That's not working. Dark blue, this area in here. And these are known as vestibular folds, or also known as false vocal cords. False vocal cords, you know, they can help close the epiglottis or close that airway a little bit tighter, but they don't have a function as far as sound. So the vocal cords, these guys, the light blue ones, have a lot of elastic fiber, but also ligaments, and they vibrate. So the job of them is to produce sound, and as the sound goes up, you know, is forced out, causes vibrations of these. How loud it is depends on the force of the air. The pitch of it depends on the vibrations of those fibers, or those ligaments, and how long those are. So the longer the ligaments, the more deep sounds that you're going to hear. You know, manlier voice, I guess you could say, or deeper voice. The Adam's apple protrudes out further. There's more sort of space there for longer vocal cords. Longer vocal cords lead to deeper sounds. Little kids have little tiny, cute little voices because they have little vocal cords. So I believe the next slide discusses the things that I just said, so we'll go over them again. All right, so what I just said was this. Speech is from expired air, so being forced out. Pitch is determined by the length and tension of the cords. Loudness depends on force of air. And then we have all of these chambers that amplify and enhance the sound. Pharynx, oral, nasal, sinus cavities, anywhere there's space, like open space. And then the last part is that sound is shaped into language by a whole bunch of muscles. All right, so our vocal cords, or vocal folds, sorry, vocal folds may act as a sphincter to prevent air passage. And I think I kind of said that earlier. So we have our vocal cords, and then we have our vocal folds. So the vocal folds, I can't say it, I'm so sorry. I recorded this earlier. It was 2 hours long, and it got deleted and didn't save. So I'm trying. All right, so your larynx, your vocal cords. Okay, so your vocal cords do their vibration to make sound. That's not what I'm referring to. What I'm referring to are the vocal folds along the outside of it. And the vocal folds can help close that airway a little bit more during, you know, if you're really trying to close that airway off quite a bit. There's a maneuver called the Valsalva's maneuver. This maneuver is done, it's really just closing off that airway, forcing pressure in your abdomen. So it's kind of doing a sort of motion, but you're closing off your airway and you're sort of tightening your abdominal muscles. So this would be used, you know, if you're trying to push something out of the uterus or the rectum, or if you're lifting something very heavy, it helps kind of activate those abdominal muscles. All right, here's our tracheal structure slide. This is slide 18, and I'm looking at, this picture is not in your textbook, but the tracheal stuff starts on 827. Yeah, slide 18, sorry. Okay, so again, if you're doing your steps, we had number 1. Oh, why does he keep doing that? Number 1, nasal cavity. Number 2, pharynx. Number 3, larynx. Number 4, trachea. From there, after trachea, we're going to go to bronchi, then our bronchial our different bronchi. Our bronchioles eventually will go down into our alveoli. All right, I think my highlighter will actually work really well with this slide. As long as I don't leave it black. Okay, here we go. So the trachea is known as the windpipe. So it's going to be extending from the end of the larynx all the way down to about here, to right here, to the branching location. And what we have are C-shaped rings, and those rings are made of hyaline cartilage. So again, this is hyaline. This is so hard. I wonder if I can do it with my finger. Oh, I can. Sorry, I didn't know that. Okay, so hyaline cartilage. I'm going to erase that and do it again. Not all of this is hyaline cartilage, right? So it's just the rings. And the rest of it is mainly muscle. Okay. And so these rings are C-shaped. So if you look at it from the front, we see kind of this shape. If you look at it from the back, you'd expect it to kind of go all the way around, but it doesn't. Instead, it goes like this. The back is made of a different material. It's more muscle. And so we call these rings C-shaped because, and the next slide will help reiterate that a little bit more, but because they don't go all the way around. So now butting right up against the back of the trachea. So if I had, this is the trachea, and then I had, this is the esophagus. All right, so if we're looking at this as a sagittal view, right, and so that would make this the front and this posterior. If food moves its way down this way, if we happen to have a big mess of food that got swallowed at once, it's going to smush into the trachea. And so in order to prevent that from happening, or in order to prevent it from bumping into something really solid, the back side of the trachea is softer. And so the esophagus, even though it's right up against the trachea, it can push against it, but two things that are sort of smushing against each other is a lot less damaging than something smushing into something that's all the way hard. And so these rings that are part of the trachea are C-shaped. They don't close in the back. So then the last thing I wanted to discuss with you on this slide is this region right in here, which is known as the carina. So the carina is just literally a spot, a position, a location where the trachea ends. And when people do an intubation where they'd have to, trach someone, they put a tube in and they aim for it to stop right around at the carina. They don't want it going further than the carina. If it goes further than the carina, it tends to go right down here. And that's not good because then this lung over here is not happy. It's not getting anything. And so we'll discuss a little bit more about that in a little while. All right, so there's your trachea and then the carina, and our next step is going to be the bronchi. Oh, so we're not ready for another step yet. I apologize. All right, and so let's find this. Okay, so this structure in the back here is the esophagus. Notice there's no cartilage, there's no bone. This is all muscly and stratified epithelium. And so this section right here, I'll do it in green. This is the back of the trachea, the posterior side of it. Notice that the cartilage is this blue, does not close this whole area off. Muscle closes it off. So that this surface and this surface, when they touch, they will not damage each other. Also of note, on the inside of this is the lumen of the trachea in here. Oh my gosh, sorry about that. The lumen of the trachea in here Notice all these little fuzzy cilia along the edges. They're going to be responsible for helping to filter some of that waste out of the air that's coming in. And also hike that mucus out of here. All right, next step, bronchi. All right, we had our trachea, we had our carina right in there. So trachea and carina, check. So the next thing is we need to discuss where the air is going to go from there. So once it passes the carina, the air has two choices. It's either going to go to the left or to the right. If it decides it's going to go to the right, then the pathway in is kind of easier because it's more horizontal, more vertical. So it kind of just continues on going straight. The left side kind of bumps out quite a bit. It's going to be much more horizontal. The reason being we have a little heart that sits here. And the heart smushes this way a little bit. So what we have in this area is a structure called the cardiac notch. Right here. Cardiac notch. And also what's going on here is therefore there's a smushing going on of this left lung, and we end up having this bronchus kind of have to go up and over it. And so the right main bronchus, this guy, is wider and shorter and more vertical. The left one is a little bit skinnier and longer and more horizontal. So now as far as the lobes of the lungs, the right side actually has 3 lobes and the left side has 2 because of that heart. The bronchi are going to branch, but the number of branches that we have is dependent on the number of lobes. So you're going to see, well, you'll see in a second. So let's go to the next slide. So this slide is slide 20. And this picture is on page 828. This is a really awesome picture that one of my colleagues sent to me at one point. But this is just awesome. So we're looking at like a cast. We call it a cast of the airways in the lungs. So you're looking at the carina right in this area here, and then we have our left primary bronchus and our right primary bronchus. So your primary bronchus is your first branch. Once we have that branch, it's going to branch into a secondary branch, and then those branch into a tertiary branch. So the secondary branch is known as a lobar branch, and then the tertiary branch is known as a segmental branch. And as these branches become smaller and smaller and smaller, we eventually call them bronchioles. So if you remember, we had veins, then venules, then capillaries, then arterioles and arteries. So we're talking about smaller vessels and smaller structures when we use the word ole or ule. And so once we pass the primary bronchi, the secondary bronchi, the tertiary bronchi, we then go into bronchioles. And we're still conducting zone structures here, guys. We have not done any respiratory exchange or anything like that. All we're doing at this point is getting it in and getting it, heated up, moisturized, whatever. So, so our initial or our first proximal, I guess you could say, proximal bronchioles are less than 1 millimeter in diameter. So they're really, really small. And then eventually those are going to become terminal bronchioles. Terminal bronchioles are half the size of those. So we're looking at 0.5 millimeter in diameter. So very, very, very tiny. Once we get to the terminal bronchioles, now we're entering into the respiratory zone. So let's go to the next slide, which is slide 22. All right, and this is your pathway once you get to the trachea. So we had been kind of writing down, okay, number 1, nose, number 2, pharynx, and moved our way down. Once you get to the trachea, these are the steps through the end. of airflow, because once it gets to the alveoli, it will then enter the bloodstream. So looking at our structures that we already discussed, we have our hyoid bone, we have our thyroid cartilage and our cricoid cartilage. The thyroid gland we didn't really discuss, but I did a little. I said it sits kind of down here. Trachea with its C-shaped rings, carina at the base of the trachea. Excuse me, left primary bronchus, right primary bronchus. Remember, your left primary bronchus is not going to be as vertical. And then we have our secondary bronchi and our tertiary bronchi. So that's all the way through to here. And then we get to the bronchioles, terminal bronchioles, and then we're entering the respiratory zone. So here's where we're going to start, seeing the functional aspect of the lung, or of the respiratory system. I'm sorry. Okay, so respiratory zone starts at the tips of the respiratory branches. So we have our terminal bronchioles, which this, sorry, let me find. Okay, terminal, nope, okay. Go back to my yellow. Terminal bronchioles. So we're talking at the very end of all of those little bronchi. We have our bronchioles, then they become respiratory bronchioles. Respiratory bronchioles are leading out to these little air sacs. Respiratory bronchioles kind of continue. You can't see it too well, but they extend to become alveolar ducts. And those alveolar ducts lead to alveolar sacs, which lead to alveoli. So the alveolar sacs are clusters of circular, they're almost like balls, but they're all connected to each other and they're hollow. They're connected to each other through pores, but they do have walls. And there are about 300 million of them. But this is where the actual gas exchange is going to happen. These alveoli are delicate. They're simple squamous epithelium, which, is kind of a negative because they're delicate, but it's also a positive in that gas exchange can happen really rapidly through the walls of the alveoli. Oh, this picture is on 829. All right, and now we have, this slide is on page 830. This picture is on 830. And so talking about this respiratory membrane, we have our terminal bronchiole, our respiratory bronchiole, and at this point we have a lot of smooth muscle involved. And so the autonomic nervous system can have like a big role on, excuse me, lung capacity. People with asthma, we'll talk about this actually in a little bit. As far as what controls the diameter of these. Let me move myself here. I don't know if I even can. Hope I can't. Anyway, all right, so smooth muscle. All right, so we're talking about our terminal and respiratory bronchioles, right? And so we brought the air in, and we are entering into these alveoli. If you were to cut an alveolar sac open, this is what you would see. It looks like the inside of a wasp nest. We see all these little compartments that have walls, but they also have holes in them to connect to the other ones. So this whole alveolus might kind of expand and recoil at the same time. Surrounding that is going to be all of these capillaries. And these capillaries are also one cell thick, simple squamous epithelium. So the exchange of gases, oxygen in, carbon dioxide out, is very, very rapid. So the alveolar walls we already talked about. So there are a couple kinds of cells in the alveolar walls. There's type 1 and type 2. The type 1 cells are, the squamous cells. Type 2 cells are a type of cuboidal cell that secretes something called surfactant. Surfactant is a chemical that aids in, it prevents these alveoli from collapsing with every breath. All right, and so this is slide 25, and this picture is on 830. All right. So elastic fibers are part of this whole thing. Again, it kind of expands and recoils together. These little holes that I'm trying to highlight right here are alveolar pores, and that allows these guys to connect to each other. Again, we're looking at a frontal section, I guess you could say, of this mass of these alveolar balls, I guess. So those little guys, these are the alveolar pores, and they help keep the pressure equal throughout. These green guys are the surfactant makers. We'll talk about that a little later, but that was your type 2 ones. And then the purple ones, these guys are macrophages. So we had said a couple lectures ago that, oops, I really don't need to color all of these in. It's just something I feel like doing right now because there's the whole coronavirus thing. Okay, anyway, sorry about that. So these purple little guys are macrophages. Those macrophages were once monocytes. And they left the tissue, and they went into the lungs and decided that's where they were going to live. So these macrophages are constantly fighting battles with bacteria and germs that try to make their way in. And so a lot of times they die in this battle, or they'll ingest something and die as well. And so they get kind of coughed up. We have those cilia that are going to sweep this up, and we cough them out, or we swallow them, actually, up to 2 million per hour, which is disgusting. But that's it. So then over here, so over here, then we have a depiction of what's happening at this boundary between the alveolus and the capillary. And you can see the big take-home message with all of this is that we want to have gases exchange, oxygen into the capillary, carbon dioxide out of the capillary, into the alveolus, and then out of the body. This happens quickly, very quickly, because of the thickness of the walls. So then we have surfactant. Surfactant is, I'll put it in green because that's what the cells were, that green color. Surfactant is something we'll discuss a little bit later, but it's a substance that is secreted into the fluid coating the alveolar surfaces. So you have an alveolus, and the inside of it is kind of coated with this, oh, that was supposed to be blue, surfactant. And so whenever this when this alveolus expands or recoils, it's not going to collapse. Without surfactant, this recoiling just leads to a pretty much deflated alveolus. So the surfactant helps prevent that from happening. Sometimes it doesn't start until 2 months prior to birth because the lungs are not functional. And so if a fetus is born too early, a premature baby, they might have something called respiratory distress syndrome. And pretty much every breath, their alveoli are collapsing because of that lack of surfactant. All right, this is our lungs. Our lung is our kind of our last topic. So if you did your pathway and you wrote it all out, the lungs are not the last stop in the pathway, I guess, but the lungs are pretty much all of these structures, primarily alveoli. So the lungs are like made of all of these. It's not like its own stop, if that makes sense, as one of your numbers. All right, and so a few of the terms that we need to go with, or we need to understand about the lungs. First thing is that the lungs fill up the whole thoracic cavity with the exception of the mediastinal structures. So we're talking about the heart, the major blood vessels, all the structures in here are mediastinum, the rest is all thoracic cavity. As far as terms to know, the root of the lung is the medial site of vascular and bronchial attachment to the mediastinum. So it really just means where the bronchi and blood vessels, major blood vessels are going to attach. And then the costal surface of the lung is the outer surfaces that are going to come in contact with the ribs, because costal meant rib. So yeah, mainly alveoli. Besides alveoli, the remaining, that is not what I wanted to do. The remaining tissue type is stroma. Stroma. Stroma is almost like a scaffolding. It's made mostly of elastic tissue. So we got alveoli and stroma being the major components of this soft part. This is a good diagram. It's very sort of simple. Kind of shows you all of these main structures. Similar to 832. And then I wanted to point out, while I have my book right here in front of me, table 22.1 and 22.2 is a nice summary of the structure, a picture of the structure, description and features of the structure, and function of the structure as far as, you know, your respiratory system, upper respiratory and lower respiratory division. So those are good. Those are good study guides. I would look at those. Anyway. Excuse me, I don't have coronavirus. I'm fine. I have allergies and I've been talking for a long time. Okay, so structures to the lungs to know. Let's kind of review again. I guess you can't get enough of review. Thyroid cartilage, cricoid cartilage, trachea, the hilum. Okay, so the apex of the lungs is the pointiest part, which is at the top. On the heart, the apex was at the bottom-ish, but apex just means the pointiest part. So on the lung, the pointiest part happens to be the apex. The base of the lung is along the bottom. This is where the diaphragm is going to sit as well. So then we have the hilum right here. And a hilum is a location where similar to the root, but this is where things actually plug into the lung. I don't have a picture of that at the moment, but in your book on page 832, letter B, you're going to see, you'll see that labeled hilum. So it's the location where the bronchial tube, the pulmonary artery, pulmonary veins are going to go disappear kind of into the lung tissue. All right, so the apex, the base, the hilum, and then we have our left lung, which we already kind of discussed, and we said that there's a cardiac notch kind of built in right here. It's actually kind of like this. So cardiac notch, remember we have an upper lobe and a lower lobe. Upper lobe, lower lobe. And we have fissures. So the oblique fissure, I'm going to do this in a different color, orange. Oblique fissure right here divides upper and lower. And the other side has two fissures because the right lung has 3 lobes. So horizontal fissure here and oblique fissure here. So our fissures are, and if you scroll back a few slides, or one slide actually, sorry, to the real lung, the actual lung, you can see that these lobes are separate from each other. They're connected posteriorly quite a bit, but not all the way through. And so the fissure is just kind of like the crack where we're seeing the difference between them. All right, and this is our picture that we have on 832. So again, just another image, you know, showing us those structures. We're not doing this branch. I'm sorry, we're not doing this slide, so you can skip this one. And here is our discussion of blood supply. So we're going to do blood supply, and then we're going to do pleurae, which would be the membranes that sit on the lung and line the lung cavity. And then once we're done with those, we're going to be entering into the physiology of the lung. So let's look at our blood supply for a second. Pulmonary arteries and pulmonary veins, we've already discussed. We know that the pulmonary arteries are bringing deoxygenated blood. So pulmonary arteries are bringing blood from the right side of the heart and bringing it to the lungs to be oxygenated. And those particular vessels branch, end up becoming those pulmonary capillary networks that surround the alveoli. And then the pulmonary veins are going to carry that oxygenated blood from those capillaries and return to the heart to be pumped to the left, the left side of the heart. So our last thing here on anatomy would be the pleury. Okay, so on this particular picture, you can see the diaphragm is this guy down below, diaphragm, and then we have the mediastinum is this area in the middle, and then we have our ribs along the outside. And so the thoracic cavity, the rest of the thoracic cavity, we have the lungs and we have the linings that are involved. And so I don't have pink. I wanted to have pink, but the pink one, the innermost lining, is known as the visceral pleura. So remember, the visceral pleura is a serous membrane that's going to sit on the surface of the lung. Pleura means lung, visceral means that it's a serous membrane that sits on the surface. And then the blue one, which is kind of outer, is the parietal pleura. You can't see that at all, can you? Nope, sorry about that. Here we go. Parietal pleura lines that cavity. And then in between the two of them, there's a white space. I thought I could draw with white. Looks like I can't. But anyway, there's a white space, and that is called the pleural space or pleural cavity. It's like right in there. So these membranes all are going to contain serous fluid. They're going to make serous fluid. And that serous fluid is slippery, and it coats these surfaces because the lungs are constantly moving, and we do not need friction. And any kind of friction might lead to something called pleuritis or pleurisy. It can cause scar tissue. It can cause all kinds of things. It could cause, scar tissue to form and kind of build up in this region and fuse to the diaphragm. Like, it can be a big deal. So those are your pleury. And so this is going to kind of conclude the anatomy portion of this PowerPoint. And the second half of this, slides 33 to 60, will be the physiology aspect. So yeah, so if you're ready to go on, then keep going. Okay, I'm going to keep going. All right, here's our respiratory physiology, ventilation. So the oh, sorry, I thought mine wasn't working. All right. Sorry about that. Okay, so respiratory physiology. So the first step in respiratory physiology is the ventilation. Excuse me. Ventilation is literally breathing. It's getting air in and out of the lungs. So we use the word inspiration for breathing in. We use the word expiration for breathing out. And it depends on pressure changes. Volume changes causes pressure changes within the thoracic cavity. So we'll discuss how that's done. It's really mainly by muscles. Diaphragm and intercostals are the big players here. So there's a special law called Boyle's Law. Boyle's Law states that if volume of a container increases, pressure decreases, and vice versa. If volume decreases, pressure increases. So picturing the lungs as being balloons, which they really aren't, but let's picture them being a balloon. Excuse me. As that balloon, let's say that balloon suddenly enlarges. What's going to happen is that the air inside now has more room to spread out. And so that's going to lead to the ability for air to rush in because we're making more space inside. As more air rushes in, that happens because the pressure has dropped. What happens with our lungs is that we have our lungs doing their thing. They're just sitting there. And there's a muscle underneath of the lungs called the diaphragm. And there are muscles all around the lungs, which are the intercostals. And this is your lung. When you drop your diaphragm, which is like this, when you drop that lower, you're actually taking your lungs and you're making them taller. And your intercostal muscles, your external intercostals in particular, lift the rib cage out. And so you're not only making it wider, but you're making it longer. From the shape you had, you've gone this way and this way. So volume has increased. And that means pressure has decreased and air rushes in. Now, if my internal intercostals squeeze and my diaphragm goes back up, now I have this larger cavity that's going to shrink back down again. Air is going to get forced out the top because pressure will increase. So Boyle's law, discussion on 835 of pressure. I think that pretty much covered this slide. So yeah, we call that a pressure gradient, and it's just like diffusion. This gases and really materials want to go from where it's more concentrated to where it's less concentrated in general. That's the goal. That's what they want to do. So this doesn't require energy other than the muscles. So that is energy, but the contracting of the diaphragm and then the contracting of the external intercostals. So your external intercostal muscles allow you to breathe in better, and your internal intercostals help you breathe out better by squeezing. So internal intercostals squeeze, external intercostals pull out. And so that's I just gave you an overview of these slides. Inspiration is an active process, and that means that it's going to take some contraction. It's going to take some movement. Active process. So during inspiration, the dome-shaped diaphragm flattens as it contracts. So it's going to be here normally, and when you inspire or breathe in, it's going to go flat, and that means that the lung will be able to get taller. The thoracic cavity is going to go up. And then, of course, the intercostal muscles, the external intercostals, are going to raise the ribs up, and it makes it wider. And so the whole shape of the thoracic cavity changes from here to here just by the contraction of those two muscle groups. Now, expiration is actually passive. Passive. That means that it really doesn't require normal respiration, normal expiration, normal breathing out. Doesn't require it doesn't require any muscles to contract. It requires them to relax. And so the diaphragm relaxes, the intercostals relax, and it changes the shape again so that the air is forced out. And then once we contract those muscles again, then the volume changes again, air rushes in. And it rushes in because there's a pressure gradient or pressure difference. So in summary, we have to use energy during inspiration to contract those breathing muscles. Expiration is passive, the muscles relax. If you are forcing an expiration, like if you're trying to breathe out, like you're breathing your bad breath on someone, like smell my breath, that kind of thing, or you're stuck in the house, that kind of thing, forcing it out uses muscles. It uses abdominal muscles. It also uses your internal intercostal muscles, depending on how much you're forcing. Pectoral muscles can also become involved with like breathing as well. But yeah. Okay. All right, I'm on slide 38 right now. Physical factors influencing pulmonary ventilation. So there are three things that are going to have an impact on how the air flows. And it's kind of similar in a way to blood pressure when we talked about vasculature. So one thing is going to be airway resistance. Is there any resistance in the airway? There's not supposed to be, but sometimes when there's mucus or things like that inside, there can be obstructions, blockages, causing blockage. Alveolar surface tension, we already kind of discussed it. If you don't have that surfactant secretion surrounding the, coating inside, then we can have an inability for these alveoli to work properly. And then compliance. So lung compliance, we'll talk about that in a second. All right, so airway resistance. There should not be any friction. We do tend to see friction, like I said, if there's mucus or some kind of infection. So once the air gets to the terminal bronchioles, so remember we had our, our pathway, our bronchi, our primary bronchi, secondary bronchi, tertiary bronchi, bronchioles. We end up getting to terminal bronchioles and then respiratory bronchioles. So at the terminal bronchioles, we end up kind of having no more resistance, because at that point, from that point forward, we're doing gas exchange. And so the actual movement of the air at that point, or the chemicals in the air at that point, is dependent upon diffusion. And so the air is going to stop for a minute there, and not for a minute, but for a little bit, so that we can do that exchange. All right, so oops. I guess I can't go back. All right, I'm not sure if I skipped a slide. I think I did. So I'm just going to discuss the slide prior right now. And so that's the next slide after airway resistance with alveolar surface tension. And so surfactant is produced with deep breathing. That's when we start to produce it. And so, we don't have that developed early on as a fetus. So yes, so if a fetus is born premature, I think I did say this already, 8 weeks prior to 8 weeks of gestation, then they might need to be put on a respirator, to assist, because their lungs, their alveoli will not function properly. They'll collapse with each breath. Okay, so as far as lung compliance, compliance is a term that means stretchiness. As we get older, our lung capacities decline rapidly. Any pulmonary function tests, you'll see lower lung volumes as you get older. And that's because your compliance declines, or your stretchiness, or the ability of your lungs to expand. So the measure of change in lung volumes that occurs with given change in transpulmonary pressure. So the higher your compliance, the easier it is to expand the lungs, which means that you're going to have more lung volume. Ability to have more lung volume. So what would diminish this besides age? What's going to diminish lung compliance? Obviously, damage to the lung. A lot of infections, the fibrosis of the lung. Like right now, like there's craziness in the world as far as this COVID-19 virus, and people are having a really difficult time. Obviously, people are dying, but there's a lot of talk about fibrosis and holding your breath, things like that. We're not going to get into that in this video because it might not apply to my future students, but at any rate, so lung compliance is also dependent on things that you might breathe in, whether there's damage to physical structures, whether there's nerve damage, lots of things. But really, age is something that's common to everyone, difficulty stretching those lungs. All right, I am on slide 42 at this point, and I'm looking, all those pictures that went with inspiration and expiration are on page 837. A little bit of discussion about airway resistance and surface tension on 838, 839. Lung compliance is 840. The respiratory volumes section on page 840, 841 is going to be covered in a separate PowerPoint. And so I'm on page 844 at this point, which is external respiration. And so external respiration involves exchange of gases at the respiratory membrane. So we're talking now about, it's called external respiration. Let's just start here. It's called external respiration because we're taking gases from the lung, from the lung and putting it into the blood. So we're taking oxygen from the lung and putting it into the bloodstream. And we're taking carbon dioxide from the bloodstream and putting it in the lung. The lung is then going to breathe that out externally. And so that's why we call it external exchange. So oxygen diffuses from the lungs into the blood. Carbon dioxide diffuses from the blood into the lung and then out of the body, external. There's a little video clip here on external respiration. If you go back, and I think you should be able to click that. Yes, if you're looking at this slide by slide instead of as a show. This slide, you can kind of ignore the top section here, but down at the bottom, we have our, sort of a similar slide to what we were looking at before as far as our cells involved. These little purple guys, again, are our surfactant makers, and the little green guys are the, nope, I said it backwards. Erase. Surfactant secretor, they look kind of fuzzy, that's why. But anyway, the purple ones here are your macrophages. These are the guys who we're going to try to clean up. And then we have the green ones are our surfactant secretors. So what's happening in this right here is happening right in here. And so what's going on here is the left side here, this is the alveolus, the inside of the alveoli. And then over here, this is the bloodstream. This is the blood plasma and bloodstream. And so carbon dioxide wants to diffuse out of the bloodstream into the lung or alveoli. This is a mess. Carbon dioxide wants to go out of the bloodstream. Oxygen wants to go into the bloodstream. That is not because of what it wants to do. It's because of diffusion. And as diffusion is that substances want to go from where there's a lot to where there's less. So this naturally happens. But then once the carbon dioxide has gotten to the alveoli, we then breathe it out. We off-gas it. We send it out of the body. And so that's external gas exchange. So it's happening between the bloodstream and the actual lung. All right, this is slide 44. We're almost done actually, so hang in there. So ventilation perfusion coupling, this is something that I'm really quickly, I'm not going to go into a lot. It's page 845. It's a very detailed topic that I don't have tons of time for with you, but it is something that you might discuss if you go into respiratory therapy or something like that, as far as partial pressures and things like that. But the terms perfusion refers to blood flow. Oops. Perfusion refers to blood flow. Right here. Fusion, blood flow. Ventilation involves gas. So ventilation and perfusion lead to that exchange that I just mentioned. All right, so internal respiration is sort of the same thing. We're still exchanging gases, and actually the slide on the next page is the next slide is it has a picture that I want to show you. So in internal gas exchange, we exchange gases again between the capillaries, but this time the exchange is between the body tissues and the capillaries. So the body tissues, we're talking about inside the body, and the blood we're talking about is the blood that picked up oxygen from the lung. So this exchange is happening between the bloodstream and the tissues. It has nothing to do with the lungs that are breathing things out. So because all of this is happening inside the body, bloodstream, body tissues, that is all internal. We call it internal respiration. In this case, we're dropping off oxygen and picking up CO2 instead of dropping off CO2 and picking up oxygen. So the next slide will help. Here it is. All right. So this whole area in the middle here, this is our bloodstream. This bloodstream is carrying oxygen and carbon dioxide. Okay, so then this area over here, this is all the lung. This is all air right here. And then over here, this is our cell. So this IR and ER refers to internal respiration and external respiration. So what is happening in this left-hand picture is that carbon dioxide is coming from the bloodstream and it's being forced into the alveolus of the lung through diffusion, and then from there it's breathing out of the body. So this is external respiration. What's coming in is oxygen. So then in internal respiration, our oxygen is unloading into the cell and our carbon dioxide is being dropped off. All right, so how is oxygen transported? So the rest of this focuses on oxygen transport, carbon dioxide, and how, what we do with carbon dioxide in the body, how it's transported and how we use it to our benefit. And then we'll talk about who controls it, who's in charge of our respiration and how we handle breathing rate, depth of breathing, how we detect the amount of gases that are in the blood and what needs to be done in the body to regulate that. So gases are transported between the lungs and the body tissue via blood. That's the slide prior that we just did. Oxygen is carried in the blood. So most of the oxygen is carried with hemoglobin. So hemoglobin can be fully saturated. That means it's 100% saturated. That means all the heme groups are carrying oxygen. The hemoglobin is considered to be partially, or that it's considered partially saturated if not all of the heme groups are carrying oxygen. So when we talk about saturation, that's what we're talking about. The next slide will help with that a little bit. So oxygen is carried in blood, like most of it is bound to iron. Fe is iron of hemoglobin. A little tiny bit is dissolved in plasma. So the opposite is true actually about carbon dioxide. So let's go there now. Oh, next slide is carbon dioxide, sorry. All right, so oxygenated blood travels via arteries. We know that, except for the pulmonary, that darn pulmonary circulation. It's supposed to be a little arrow. All right, so arterial blood, or blood that has oxygenation for the most part, is 97% saturated. It's not usually 100% saturated normally. So arterial blood is 97% saturated. Venous blood, which we call deoxygenated blood, is not actually completely deoxygenated. The arterial blood can only drop off about 25% of its oxygen during one circuit of the body. So venous blood is not going to be like 0% saturated. The body's not that effective at teasing that apart. So the pulmonary arteries and the systemic veins are going to have about 70% saturation for the hemoglobin molecules. So yeah, again, the difference is there. It's just not what you originally expect. You expect it to be completely deoxygenated. All right, so what happens if you have a homeostatic imbalance? What happens if there's not enough oxygen to the tissues? Oops. All right, so one thing that you get is this guy, cyanosis. Cyano means blue. And so you see a blue tinge to the lips, to the nail beds, to the mucous membranes. And we use this word hypoxia, hypo, which is not enough, and oxia refers to oxygen, so not enough oxygen. What could be causing this? Inadequate oxygen delivery. Okay, so part of the problem could be anemia. The anemia could be just from not enough red blood cells. It could be from something destroying the red blood cells. It could be from lack of hemoglobin, which could be from lack of iron. It could be because you have a blockage to a certain tissue. It could be from metals, toxins, pneumonias. And then we have carbon monoxide. Carbon monoxide is extremely dangerous. It's much more likely that if carbon monoxide is present with oxygen, carbon monoxide will bind to the hemoglobin better than the oxygen will, or faster than the oxygen will. And so the hemoglobin groups get all filled up with carbon monoxide and there's no room for oxygen. And so, yeah, basically that's it. So yeah, inadequate oxygen delivery, very, very dangerous. All right, so how is carbon dioxide transported? So we already know, excuse me for a second. So we already know that oxygen is transported 98 point something percent through hemoglobin, a little tiny bit dissolved in plasma, maybe 1 or 2%. Carbon dioxide is a little bit different. Maybe 10% is dissolved in the plasma. 20% is bound to hemoglobin. It's not bound to the heme, it's bound to the globin, part of hemoglobin. And when it is bound to that, we call it carbaminohemoglobin. So you need to make sure you know that when carbon dioxide is bound to hemoglobin, or the globin part of hemoglobin, it's called carbaminohemoglobin. And then 70% of it, most of it, is transported as bicarbonate ions. Bicarbonate is HCO3. And that is something that just kind of floats around in your bloodstream, in your plasma. Sorry, give me 1 second. Okay, so it floats around in your plasma. This is on page 850, like how carbon dioxide makes its way around. So bicarbonate ions in plasma. Most carbon dioxide molecules entering the plasma quickly enter red blood cells. These reactions convert carbon dioxide to bicarbonate. All right, so there is a system called the carbonic acid bicarbonate buffer system. When you're talking about a buffer, it involves pH. Buffers are things that prevent extreme shifts in pH. So the carbonic acid bicarbonate buffer system resists changes in blood pH. Your blood pH has to stay between 7.35 and 7.45 always. That's what it has to stay at. So if the hydrogen ion concentration in blood rises, remember, a lot of hydrogen leads to a lower pH, which means it's going to be very acidic. So if hydrogen ion concentration in blood rises, excess hydrogen is removed by combining it with HCO3 to make H2CO3. So what that means is we're taking bicarbonate and it's being converted to carbonic acid. So this excess hydrogen is combined with this HCO3, and so now we have H2 over here. And when it's part of a molecule, it's not going to affect the pH. If the hydrogen ion concentration begins to drop, that means the blood's going to start to become too alkaline or too high of a number. If that's the case, then this will dissociate, releasing the hydrogen ions. So what we call that, we call it an alkaline reserve. It means that it can, it's a buffer, it serves as a buffer. It can change the pH as you need it. And this is on page 851. So other things that have an influence on the carbon dioxide and pH, or really pH. If breathing is slow and shallow, This allows carbon dioxide to build up in the bloodstream, and it's going to lead the blood pH to drop and become more acidic. And so, our body has ways of trying to buffer that and compensate for that. Deep, rapid breathing pushes carbon dioxide out, and blood pH can increase too much and become more alkaline. So we do have, our breathing can have a huge impact on the pH of our blood. So we have this carbonic acid bicarbonate buffer system. We also have the urinary system that's involved with helping regulate pH. But it all has to do with the hydrogen ions. All right, so the way that respiration is controlled is neural, brain. Sections of the brain stem, the medulla and the pons are both, there's clusters of neurons in those locations that are going to be integral parts of this. So in the medulla, which is just superior to the anatomical start of the spinal cord, so we're talking about foramen magnum, just superior to foramen magnum level, there are clusters called the ventral respiratory group and the dorsal respiratory group, and those are VRG and DRG. The pons also has pontine group, which we'll talk about in a minute, I think. Yeah. Okay. So the pons and the medulla, so these key players here, medulla and pons, respond to signals from chemoreceptors. So chemoreceptors are going to detect chemicals. And the chemicals we're talking about here are oxygen and carbon dioxide. Those are considered to be chemicals. So then there are some stretch receptors that are present that are going to have a role. So all of this feedback, how much oxygen is there, how much carbon dioxide is there, how much are the lungs expanding and recoiling. These are all things that need to be received and interpreted in order for the brain to know what to do at that point. All right, so your ventral respiratory group, it's almost like the pacemaker. Pacemaker is what I was saying. Hold on. So the ventral respiratory group sets eupnea, E-U-P-N-E-A. Sorry about that. Okay, so EU. Nope, that's definitely not. Okay, EU means good or true or something along that line. So good, PNEA is breathing. So good breathing, normal breathing, 12 to 15 breaths per minute. I've seen 12 to 20 breaths per minute. If you're, an infant, it's more. That's variable, but really shouldn't be any less than 12 or more than 20 under normal breathing. So that's the ventral respiratory group is what's going to set that. Its inspiratory neurons excite inspiratory muscles via the phrenic nerve. So the phrenic nerve has to go to the diaphragm. That's what phrenic always means. Anything P-H-R-E-N. Phren goes with diaphragm. So I always remember that P-H, phragm, and phrenic. So the inspiratory neurons in the ventral respiratory group are going to send out signals, sending them to the phrenic nerve, which is going to go to the diaphragm underneath of the lungs, and then to the intercostals, which are going to be around the sides of the lungs. And so these together are going to drop that down, raise the rib cage out, and this looks nothing like what it should, but then we enlarge that chest cavity. So I'm erasing this because it's a monstrosity. Okay. So again, it's just gonna, send signal out through these nerves, which is gonna allow for the chest to expand and allow air to go in. So ventral respiratory group suppression can occur from drugs. Or certain overdoses of certain things. So what that's going to do is it's going to cause the diaphragm to not get signals. And, and that's it. So the dorsal respiratory group is gonna integrate, it basically sends its stuff to the ventral respiratory group. So dorsal respiratory group looks at peripheral stretch receptors, chemoreceptors, and it sends that information to the ventral respiratory group. So we'll talk about where these receptors are in a couple slides. So dorsal respiratory group gathers input, sends it to the ventral respiratory group. Pontine section also sends to the ventral respiratory group. So the ventral respiratory group is going to be the one to send out signals, mostly. All right, so this is what we're looking at. Up here, you have the pontine respiratory center, dorsal respiratory group, and then here's our key player here, the ventral respiratory group. And then we have the signal going out to the phrenic nerve to the diaphragm and the intercostal nerve to the external intercostals. So the pontine respiratory center is, again, that most superior center there on the brainstem. Have input toward the ventral respiratory group. This group can act to fine-tune the ventral respiratory group. So the ventral respiratory group sets the rhythm. And then small changes that need to happen can be sent from the pontine center. So in other words, if you're playing an instrument or, kind of doing something different with your breathing, whether it's exercising or sleeping or when you're talking, doing yoga, like whatever you might be doing that's kind of different than normal breathing, that might require some more conscious, not conscious necessarily, but more, I don't even know what's the right word here, but where you have some control over your breathing, at least to some extent. So like speaking and exercise or yoga or anything like that, you'd see pontine influence. All right, chemoreceptors. Chemoreceptors, the chemicals that we're going to detect, again, are oxygen and carbon dioxide. Okay, those are dissolved gases within the blood. So that information is going to be sent to the brain, and those are found in the carotid and aortic bodies, and that's on my next slide, I think. Yes, next slide. And then we also have stretch receptors, which we mentioned, that are going to have a role in letting us know, size of structures. So this is kind of what we just went over. So we have the pons and the medulla in this area here. And then we have our breathing control centers, which we just discussed. We have our phrenic nerve coming down to the diaphragm. We have our intercostal nerves going to the intercostal muscles. And then we have these peripheral chemoreceptors. They're in the carotid arteries. And that's not the carotid arteries. Oop. That's in the carotids. Yeah, that's kind of weird. Yeah, no, I'm sorry. This is, and this is, yes. And then the aorta. So we have our chemoreceptors. Your book doesn't go into too much about those stretch receptors, but there are stretch receptors in the lungs. Yeah, to let you know, to let the body know what's going on in the lungs. There's sensors. All right, so now a couple other terms. This guy right here, hypercapnia. So capno means CO2, and that means excessive carbon dioxide. Hyper is excessive. Okay, so if carbon dioxide levels in blood are too high, carbon dioxide is going to accumulate, and it can accumulate in the brain. If it accumulates in the brain, we have pH drop happening. Low pH is going to stimulate the brainstem chemoreceptors. That in turn is going to increase the depth and rate of breathing, which is going to then lower the carbon dioxide and change the pH again. So if carbon dioxide changes, this whole series of steps happen, pH will drop, and then the central chemoreceptors, the chemoreceptors in the brainstem, are going to detect that drop, or detect that drop in pH but increase in CO2. So they're going to what they're going to detect is the carbon dioxide, and so then from that point, it's like a switch, and this process starts to change your breathing and decrease carbon dioxide in the blood, which is going to increase your pH to where it needs to be. So we have ways of controlling pH through our bicarbonate buffer system and through breathing. All right, we're almost there. We've got 2 slides left. All right, so a few other definition terms. We have hyperventilation. Hyperventilation, excessive breathing, increased depth and rate of breathing that exceeds the body's need to remove CO2. So when you think about someone who's hyperventilating, they're doing that. So when you're breathing out really fast, like that, you are you're removing too much carbon dioxide actually, and you're not breathing back in enough. And it's going to cause changes, dizziness, all of these things. So people have a brown paper bag that they breathe into. What they're doing is they're kind of like recycling that carbon dioxide and forcing it back into the bloodstream. And so what we would call that when the carbon dioxide is too low is hypocapnia. So remember, hypercapnia was too much. When this happens, it can lead to a lot of issues in the brain with blood vessels, and could lead to passing out. And so that's why the brown paper bag thing happens. Carbon dioxide levels can hopefully return back to normal. So apnea, and the PCO2 is just think of it as carbon dioxide concentration. So apnea is absence of breathing, stopping breathing. This would happen in sleep usually. It starts up again. So it's like, for a lot of reasons, a person might stop breathing, but then as soon as we start to see the chemoreceptors start working, we'll force the brain to wake up, and we'll force your body to wake up and gasp. It can happen hundreds of times a night. So hyperpnea, it means increased breathing. So this is increased ventilation. It can just be from exercise. It doesn't have to necessarily be from something bad, but it can be from a fever. It can be from lots of things. Most commonly, it's going to be from exercise. All right, so now the last thing is that there are controls that go with the limbic system. So remember, your limbic system was involved with emotions, and hypothalamus, the limbic system is like, it's almost like this snake almost that runs through the brain, and then it has all of these connections to it from all these different parts of the brain. So emotion can have a physiological response, and this is no exception. So breathing can be very, very easily influenced by emotions. And so the hypothalamic controls act through that limbic system, and they can cause changes based on your mood. So one thing that I do with my son all the time, who's 4, is like, chill, like calm down, like breathe. You need to breathe. You're angry, you need to breathe. Or like you're whatever 4-year-olds are upset about, I don't know, that washing your hands is boring, so there's a meltdown. You need to breathe. So when you're angry, you tend to hold your breath. Or when you're frustrated, or when you're in pain, like you do different things with your breath, and those things are not necessarily under conscious control. But recognizing it, like your emotions can change based on that as well. So if you practice slow breathing or trying to get control when you are breathing, then we have this, it's hard for me to explain, but it's your emotional center is tied with the center that's in charge of primitive drives. So that's where the hypothalamus comes in. And so I guess the point of this is that it's not just conscious control to change your breathing. It's not just the, ventral respiratory group. It's the hypothalamus and the limbic system have control over this based on, interpretation of situations. And so I already talked about body temperature increases respiratory rate. And then we do have some controls that are in the cerebral cortex. So we have our hypothalamus involved, our limbic system involved, our chemoreceptors and our pontine respiratory center and our medullary centers. So the cortical controls, remember the cortex of your brain is the outer aspect of your brain. Cortex means bark, like bark of a tree. So the cortex controls are direct signals from the cerebral motor cortex that bypass the medullary controls. So if I want to control my breathing, I can do that. And it's the same thing with, and that, in turn changes a lot of things as well to control your breathing. But in the case, let's say, of holding your breath. So you hold your breath and you're voluntarily doing that. Your cerebral cortex, the motor center of your brain, is the one who's allowing you to, take control of your diaphragm there and hold your breath. So you're going to do that, and you can do that until your brainstem central chemoreceptors are going to detect carbon dioxide levels are way, way, way higher than they should be, and it will override the cortex, and you'll take a breath. And so this is how drowning happens. The person holds their breath. Once carbon dioxide levels build up, then the brainstem is going to come in and say, you know what, we're not breathing, we have to breathe, and it will take over because that's its job. So anyway, slide 61 is actually nothing. I don't know why. I was going to put 2 links there to crash course videos. So if you look up on YouTube, crash course biology, and there's a part 1 and a part 2 of the respiratory system. Please watch those videos. One of them is 9 minutes and one of those is 10 minutes, I believe. So please watch those 2 videos, and yeah, and that's about it. So if you have any questions about this, please send me an email, and yeah, and look for more information from me in the near future. Stay safe, and I'll talk to you soon.